Welcome to the ACRC webinar. We are continuing with the webinars. And today I'm very happy to have Professor Dejan Markovic from UCLA that will talk about closed loop neuromodulation. So Dejan, thank you.
Thank you, Shahar and Masha, for the invitation, and good evening, everybody. I appreciate you spending the time to discuss closed loop neuromodulation. So today I would like to share my perspectives on next generation neurotechnology for treatment of complex network scale brain disorders. So the need for closed loop neuromodulation is exemplified on many clinical studies. Recent clinical studies in Parkinson's disease with a limited number of patients have shown that if stimulation is administered about 45% of time, the same behavioral outcome or significantly, substantially equivalent comparable outcome is achieved. And also in closed loop study at UCSF with Dr. Starr and colleagues, they have shown that if you do closed loop neuromodulation and you adaptively react, you can spend 40% less energy to treat Parkinson's disease. And of course, less energy is better because that means longer battery life. It also means smaller volume device, and also means less undesirable effects because we are injecting less energy into the body. Dr. Lozano, who is running now phase 3 clinical trial at the University of Toronto, has described the need for closed loop DBS for Alzheimer's, particularly calling for the need for phase locked stimulation when you're synchronizing with the theta phase, the stimulation that would be then coinciding with encoding or retrieval of memories for cognitive performance. And colleagues at UPenn and their associated clinical centers have done studies in closed loop DBS for memory and TBI. So there is a great need, but technology is not there yet. So there are some important technology gaps that need to be closed to fully address closed loop neuromodulation. It comes down to the need for sensing, of course, from a high number of channels that we can provide enough intervention points. Then we need to have full duplex capability to be able to talk to the brain and listen to the brain at the same time. And with sensing comes the need for real-time wireless data telemetry that from a high number of channels would implicate more than a megabit per second physical layer. And of course, with all that, competing requirement is miniaturization, adding more contact points, adding wireless, adding sensing, but making device smaller to be less invasive. So I'd like to then describe the role of technology because it's important to understand what technology capabilities exist at different time points to be able to translate that into practice and next generation technologies. And I would like to start off by going back not too far to 1973 to honor the legacy of UCLA in defining the brain-computer interface field. Professor Jacques Vidal at our computer science department introduced brain-computer interface and the term of BCI. And this illustration is a really interesting piece of history because it shows 3 layers over here with the experiment room. Subject is equipped with EEG recording equipment and then amplifiers and A to D converters. And then there is a computer area to do decoding algorithms. And then in response, we provide display to the subject so that the subject can perform 2D navigation task by moving a cursor on a computer screen. And if you look into brain-computer interfaces, this is the bread and butter and foundation of BCI, that it involves 2-dimensional navigation on a computer screen by the subjects. So you have a control area which pretty much mirrors all of the equipment in the experiment room, and that is just meant to isolate operator and the scientist from the subject as not to interfere with the experiments. There is also, in the computer area, in 1973, the IBM 360 had 4 megabytes of memory at that time, and now this is, you know, obviated by technologies that we have that are wearable and handheld. And in fact, this computer was connected to the ARPA network. This, in fact, was the first node of the internet, which came also from UCLA in 1969. So this is 2 pieces of history in this one chart, but now we have technology that's far more capable, and we have to think in a different way how we can make use of such technology features to extract benefit for society. So in brain-computer interfaces and neurotechnology, things do not happen overnight. There is a long time horizon to really responsibly roll this out into science and clinical experiments. So I'm showing here an example of 2000 researchers from Duke and MIT were able to get EEG waveforms from a monkey transferred over the internet to remotely control robotic arm at MIT Robotics Lab 600 miles away. And so this impressive experiment was enabled by the internet and BCI, the 2 fundamental technologies I described. The same group from Duke 3 years later published a study in which monkeys were controlling screen first through joystick until at some point they realized that they didn't need to use those peripherals, that they could just switch to controlling the screen with their thoughts. So that was consistent with Professor Vidal's demonstration in 1976 that a subject can, on a volition, control navigation of the cursor in 2-dimensional space that is replicated in non-human primate experiments. And there is 2017 study by Stanford University, which have shown the movement of the screen for the purpose of typing on the screen. Again, 2D navigation through BCI for a quadriplegic to be able to type on the screen. That was also a powerful demonstration in humans, although these days we have perhaps more effective technologies such as natural language processing for this specific task. Nevertheless, there is a lot of value in navigation-based tasks for human subjects. And this is the technology translated into humans. About 30 years later, from the inception of BCI, the famous Utah electrode array, which is essentially kind of a bed of needles that penetrates into cortex, motor cortex. So it goes into the somatosensory area of the brain to record single unit activity and then amplify the signals and then transfer over through a cable to a computer to decode the intent and substitute the functionality of impaired limbs for computer screen navigation. So what you can see from here is that these technologies generally require very complex hardware and software that require a significant amount of training of clinicians who need to make use of these technologies. And most importantly, it involves incredible amount of inconvenience for the subject. And that's where I like to say that those 3 things need to be always at the front of our thinking, and everything starts and ends in the clinic. That we need to come with technologies that are minimally invasive and provide meaningful functional outcome for the subject. Fast forwarding to today, this is the brain-computer interface technology by Neuralink, which has received a lot of press due to their development. This is a very impressive piece of machinery that has a lot of mechanical, electrical components for robotic implantation of a large number of electrodes in the cortex. So you can see here, these electrodes are prepared on a flexible substrate, and then this machine lifts the threads and like a sewing machine, injects them through this needle very fast at the millisecond resolution, one at a time, into high density brain-computer interface. So this is primarily technology suitable for surface of the brain, for cortical and motor function. And there is still, I think, science ongoing to find what is that we can really learn from these additional high density recordings, and how can we then introduce stimulation to make this more clinically viable. Since, access to the brain is very intimate, and there is a high bar and ethics involved in accessing the brain, which has to be justified by medical cause. And medical cause involves therapy, and that requires stimulation. So there is a lot of steps that need to be taken to make this real technology, but nevertheless, it's a very impressive development. Now, forwarding to clinical translation. So going from science, next step is from basic science to go to the clinical neuroscience and provide translation to therapy. So this is kind of a entry point for human clinical studies. That is the situation of epilepsy monitoring. Patients who are pharmacologically resistant to epilepsy and have nothing else to, no other solution than to deeply monitor the location of the seizure and track these waveforms over 2 or 3 weeks in a hospital where patients are tethered to hospital beds and this kind of clinical bulky equipment to be able to localize the seizure. And these are wall-plugged instruments, very large, with deep intracranial probes in a hospital environment, and they have very limited closed loop. And it happens that in some cases, the probes are located in areas that also overlap with other interesting translational aspects like memory or cognition. And UCLA clinicians led by Dr. Fried, who also actually works and operates in Israel, have decades of research in studying responses from the single units in human subjects. And to that extent, they've designed Benke-Fried probe, which is implanted in more than 2,000 cases so far over the past few decades, which has a whisk of microwires that are capable of recording responses from the hippocampus of single units in human subjects. So epilepsy monitoring gives us justification and ability to study science for various translational aspects. In this case, you see memory. It's also being used for study of mental health, anxiety, depression, and similar areas. And so these are the some interesting findings from the memory, that unlike motor cortex, which is kind of the BCI area, human memory is non-topographic, meaning that two memory cells that are right next to each other can respond exclusively to a completely different concepts. In motor cortex, things are very well organized and the neurons in the vicinity respond to the same function, but here it's completely different. There are, however, associations. You can have a cell responding to a sea world, to a fish and a sailboat. So that gives rise to the theory of associative learning. But nevertheless, these are very sparsely encoded, partially overlapping networks that make it very difficult to access human memory functions. And what's interesting about it is that single units, you can hear them without too much of processing, because they're fall in the audible range up to about 5 kilohertz. And so what you see here on the right, I'm going to play one video from Dr. Fried, who is showing a response from a single unit in the amygdala region from a human subject watching various movie scenes to see what the response is going to look like.
Mr. Dawson, I'm sorry, but I have the...
You can see here there is no response from this particular neuron to two people interacting in an office setting.
And how's your project coming? You still planning
Then
to
a lot
change
more
the way
people
we
in a
help
dense
scientists
environment,
view memory simulation?
in a classroom, still no response.
I gotta say that I'm enjoying adulthood for a lot of reasons, and I'll tell you reason number one. As an adult, if I want a cookie...
This neuron responds to Jerry Seinfeld pretty much and nothing else. Just however responds to Technion and the characters from the Seinfeld video to Seinfeld. And then goes again the non-responsive. So it's a highly selective to a particular concept.
And how's your project coming? You still planning to change the way we humble scientists view memory simulation? Don't we have a choice?
Alright, I'm late. I'm going to laundry. Why don't you use the machines down in the basement?
And you can see that
Puff and fold, the only way to live. I drop it off, I pick it up. It's a...
Again, no response. You get the idea. And scientists also found that this response is multimodal. Whether you see a person, read a text with a person's name, or hear, it doesn't matter which sensory input it comes from, you still activate the same cell with respect to that particular memory. Now, this is still kind of in a translational space, but looking into more mature points clinically, DBS therapy for Parkinson's disease is now very robust and has been in use for more than 15 years. So the first DBS was performed in human subjects by Dr. Benabid in France. And now it's a routine surgery that uses cardiac pacemaker technology. So this is an outdated technology from cardiac pacemakers, which is about 3 or more decades old, that is repurposed for brain stimulation by running a cable through the neck, which causes a lot of discomfort. And then you drop the lead, which has 4 contacts. So this is enough resolution to interact with the world of the brain at the level of continents. Only 4 possible clinical intervention points. But in this case, it works really well because it's a highly focal injury where you need to find the right, fine-tune the location in the basal ganglia to provide 24/7 open loop stimulation. So it's a very labor-intensive surgery. It's a 2-stage surgery that goes for 5 to 7 hours. You first put the lead in and position it in an awake surgery so that you can verify that the patient does not have tremors anymore, and then you connect the implantable pulse generator. A lot of hardware to be implanted in the chest of a person. And at UCLA, it's been performed many times. This is milestone surgery number 500 on a famous musician that showed how this works. And this was propagated through social media to increase the awareness of neurosurgery and deep brain stimulation. So you can watch this brief.
It's not brain surgery. Actually, it is brain surgery. A team of UCLA doctors implanting a pacemaker in Brad Carter's brain to treat hand tremors. But check this out. During the operation, the actor and musician was awake and he was playing the guitar. Strumming the chords actually helped guide the surgeons as they placed an electrode in his brain.
My music's really important to me, so I'm hoping that this will allow me to do that, to play guitar. I'm a finger picker. I want to record. I want to perform live again.
It's the 500th time the UCLA surgical team has performed this procedure. It's the first time that they tweeted it and had a live stream.
That is so cool.
Isn't it?
And so now we are in a situation where we are headed to cognitive disaster unless we do something radically different. So there is a number of patients who spend years trying one medication after another with no relief. And there are often cases where clinicians have nothing left to offer. And so one of the most promising approaches for that small subset of patients is to perform deep brain stimulation, or DBS. And so far, DBS have shown to be efficacious in Parkinson's disease and has very limited efficacy so far or adoption in epilepsy. So there's about 10,000 per year devices worldwide in Parkinson's and about only 300 per year in epilepsy. If you look into the prevalence in the United States, and look into depression, Alzheimer's, and chronic pain, the annual spending on managing these indications exceeds $1 trillion per year. And there is nothing that we currently have to effectively treat these indications. So the old devices have failed in limited clinical trials on major depression and others. And it really comes to the insatiable need to develop better technology. So fortunately, access to the networks of the human brain that are kind of governing the functionality and these kinds of indications are possible in epilepsy monitoring. So this is really the golden gate, the gateway to these indications where we can have ethically justifiable clinical entry point to study functional brain networks and look into the comorbidities with these various indications that would allow us to better understand translational science and therapeutic outcomes. But for that, we need technology. So if you look into the number of people worldwide with various indications and roughly which brain areas are affected, you will see that Parkinson's and treatment of deafness have been successful because the tools are at the right scale. We have enough contacts to be able to properly diagnose and intervene with the brain networks. Epilepsy, very limited, and even in Parkinson's disease, the adoption is only about 7% of all the patients who qualify. Because again, it's a very inconvenient and very bulky hardware that has to be implanted in human bodies. If you look into just depression and Alzheimer's, that alone is about 50 times more than the Parkinson's disease. But the problem is that we do not have tools that are at the right resolution to be able to achieve clinical efficacy. And it's not for the lack of trying. People have tried to treat major depression in a Broaden trial using the same low resolution technology, and that didn't work. Currently, the ongoing Alzheimer's study that I mentioned doesn't look very promising either. So common to all of these shortcomings is our inability to have the right amount of contacts, the right number of intervention points from which we can properly diagnose indication in order for us to then be able to close the loop and provide stimulation. And so imagine the situation in which we can simultaneously record multiple regions of the brain and then self-adjust therapy to provide stimulation to optimize the overall efficacy and minimize side effects. So technology is lacking, and so it's instructive to look into successful technologies of the past to understand how they translated into therapeutic world and what we can learn from them, because if we borrow on the concepts that are already established, that would lower the regulatory burden and accelerate approval. So you can see the cochlear implant here, which took several decades to be approved. It is arguably the most adopted technology just by the numbers, but it takes a decade for a subject to accept this technology after they're diagnosed. And you can see that there is this always on external technology, which does create certain social stigma about having this kind of prosthetic technology. It has a sound processor here that does the audio processing and then converts the sound into stimulation patterns that are then wirelessly transmitted over this nearly coupled coil, for which there is a need to have magnet for the alignment of the coils, just like you have in the electronic toothbrush, and then transfer power to this implant module, which does not have a battery on its own. So transfer the power and the stimulation patterns that are then applied to the auditory nerve and to the cochlea, and to stimulate the auditory nerve that goes then to the brain. Perhaps the most impressive piece of technology from the technology perspective is the retinal implant. This is the technology developed by Second Sight here in Southern California. That was FDA approved in 2013, and Second Sight implants about only 75 people per year. So there is a number of complex issues here that come to such a low adoption that comes from fairly obtrusive technology that has the battery from like VCRs from the time that FDA process initiated to power these transmit coils and the glass montage over here over the eyeglasses, which have a camera that then transfers the stimulation pattern applied to the retina in a mirrored and inverted way, that the way that this works. But if you look into the technology, it took about a decade nearly to make sure that you have the electrode array that does not have basically detachment from the retina, that can be reliably interfacing with that interface. Then it has receiver coil that is wrapped around eyeball together with the electronics case, which is a titanium based case. And you have these suture tabs that holds this in place. And now you have a constant displacement with the eyeball moving with respect to this transmit coil, where you need to provide power to this implant. So it's incredible technology, and yet you have only a handful, couple dozen electrode contacts to provide just you know, kind of a sensation of flashing lights rather than really kind of full image. There is other technologies sponsored by government. So these are some of the DARPA technology, the EEG cap, the high dexterity arm that interfaces with nervous system to provide mechanical movements of these bionic limbs. And then there is the BCI, the experiment that I mentioned with the typing, which uses this BCI technology. And like I said before, what's really common to these technologies, you can see that there are very, very complex pieces of technology, both hardware and software, that clinicians require extensive training, and they're really inconvenient for the subjects. So we need instead technologies, particularly in this day and age of miniaturization and technology advancement, that we can seamlessly embed into the fabric of daily living and to be minimally invasive. So that's the President's Brain Initiative came 2013-14, and DARPA was at the forefront of President's Brain Initiative with a couple of programs. This is the Subnets program, which aimed to develop platform technology, so R&D level, for precise closed loop therapy in humans. Government had interest in mental health issues for the military, but this could be, of course, translating to other indications in civilian populations as well. And at that time, a team of device developers, clinical researchers, and animal researchers teamed together to develop this technology. I'm obviously on the technology part of this, and I also worked with teams working in restoration of active memory to come up with a platform technology that has about 200 contacts with the brain networks, which would be radical departure from only 4 to 8 contact brain interface, that we can really address these systems network scale disorders. And so our team at UCLA, working together with UCSF and large number of collaborators, my team led the development of the technology and system integration and all of the electronics modules. And so this is the implant here, which we were able to develop, that has closed loop stimulation with up to 256 channels. You have flexibly the ability to have cortical or subcortical depth electrodes, each with up to 64 contacts, with modern technologies for wireless power and data. We've also developed PC application and user interface that allows clinicians to access the recordings and program the device for stimulation on various points. So the overall idea was to develop objective system for personalized treatment. At the implant side is the device, but that is just a starting point, the interface. Beyond that, you need to work on the data analytics and understand what kind of algorithm would work for general population, but it's specific enough that can be individually tuned. And further to the subjective tuning and involvement of subjects, there was mobile application, which was called Modify, that allows subjects to provide their feedback and subjective input into this algorithm and implantable devices. So this will be the technology that is synchronized with the functioning of the implant. And we spent 5 years developing this technology in the R&D space, more in the research domain. And you can see for those students who work on the electronics, our paradigm here was that each of the electronic modules, each of the custom chips had to be validated as a piece of the full system. So this is just the benchtop step in which you see stimulation and power management electronics with 32 channels, sensing control with microcontroller and FPGA boards and the associated software that runs this closed loop. And then this was powered with wirelessly transferred power over the closely coupled, closely spaced coils. So everything had to work in a system. And once that is proven, we can make it into a smaller scale system that can be used as an external trial system for convenience of portability and demonstrations in animal and human subjects. And once that is fully verified, then we can go to the implant scale, where we can package the stimulation and sensing into a vitamin pill sized module that can be then packaged for implantation. So we envisioned a system which has the ability to simultaneously record from multiple locations, aggregate signals to a hub, and then send them to a controller. In response, the controller would send power and therapeutic stimulation that is individualized to each patient. So this device features the full duplex and world's smallest human specification 64-channel neuromodulation. You can see here that it's assembled using medical grade components. So you have sensing and a crystal oscillator, then you have a stimulation power management, and then you have a stacks of medical grade capacitors, which were wafer scale components cut into pieces for assembly. This PCB alone was a 12-layer PCB with optical technology for traces and vias, 2 mil traces and spaces, and a lot of blind vias. And this was really fantastic experience for debugging and putting all of these stacks. So from the perspective of electronics work and miniaturization, this was a great example. And then this was then placed on ceramic substrate and assembled and laser welded in a titanium encapsulation. And then we have 6 pins for differential power, clock, ground, serial in and serial out to access all of these channels on a smart lead that fuses electronics with high density, high channel count electrode array. And this technology was made to be modular so that we can flexibly switch between cortical and subcortical. And admittedly, it could have been made with fewer components and could be placed in one module that is cranially mounted. But at the same time, we want it to be modular and to provide more flexibility as a platform. We also had a commercial partner on our team to provide enclosure for our device. So we borrowed their power charging coil and developed customized electronics with microcontroller and a custom chip for wireless communication. And then we passed all the inspection points at their lab to provide verification for qualification units for further maturation of this technology. So these are the high density electrode arrays developed by Lawrence Livermore National Lab, 64 contact arrays for both cortical and subcortical. These are 4-way splits of 16 ring contacts for current steering. So this is much more specificity that is required, really. If you look into the anatomical variation from the images of the brain, from the brain scans, you see that there are inevitable variations. And so a high number of contacts gives us sufficient coverage to choose the right and relevant contacts for each subject. So we're not using all of them, but we use the ones that are relevant and individualized for every subject. And you will see now some of this technology.
Our neuromodulation unit
here
is small in size,
in various demos that we
which
made.
makes
This
the
is
high
first
channel count
generation technology with 32 channels. So you have the traditional DBS device, which is housing a battery, and then power had to be AC transferred, which was not the most efficient way, but you cannot put DC power through the body. And then the hub, which in itself is just a signal router, which can be eliminated, and these neuromodulators. So now everything can be placed into a single module, as I will discuss later. So this is the benchtop system over here.
The stimulation output.
Signal generators, power supplies,
running the control software,
oscilloscope to measure the output.
To change
so this
stimulation
is in vitro.
parameters.
We have a reference electrode in which we put replica of a neural signal, and then we apply large-scale stimulation so that we can do recording on adjacent channels in presence of such a large stimulation so that we can still record reference signal. the electronics was so small that we needed to develop custom pogo pin based socket for really testing, because you have a high density of contacts and very little ability to access those contacts. So we put this kind of pogo pin socket to be able to do testing. All of the type of work that students don't initially like, that has to be done in order to make this a true engineering.
Is being injected.
And so you can see here, we are injecting signal into a beaker
through the large
and then
probe
applying 10 volt peak-to-peak stimulation, and are able to record that signal in presence of this stimulation, which can exhibit around 100 millivolt amplitude that required us to develop high linearity
of constant current stimulation
technology. So this is the developer version of the application that allows us to configure stimulation engines. And you can see here the amplitude configuration.
Again,
here
we do see the waveform
configuration. And
now,
then you can look into the recorded waveforms.
As we can see,
and all this fuzziness is basically stimulation artifacts and the signal superimposed on these large stimulation artifacts. We also developed the ability to provide custom stimulation waveforms where you can customize anodic and cathodic phases and provide charge balanced stimulation. So when you do that, you can see how you can change the waveform shapes, because, you know, in biology does not produce square shapes. And so we want to be able to inject harmonics, the right set of Fourier modes to be able to excite different phase-locked networks. And that was kind of the motivation to be able to experiment with waveform shapes. When you have the ability to do simultaneous stimulation recording, then you also need to deal with the artifact rejection. And so we developed adaptive signal processing framework that first calculates signal statistics as the baseline, and then we measure a response to the stimulation at the recording site so that we can then compare that with the baseline and remove the stimulation artifacts. Because what we do know is that brain is a nonlinear and time varying system. So this has to be done in an adaptive way. And so we did adaptive filtering using template detection and statistics baseline to be able to remove the artifacts. You can see here time and frequency plots that are non-ideal, and the difficulty here is that you don't have the ground truth against you are really doing this unless you do synthetic signals, but it does a fair job of cleaning artifacts in both frequency and time domain. The way that we define engineering specifications, we really come from the clinical perspective first. So this is an example showing the right entorhinal cortex of one of those epilepsy monitoring patients, in which we have a depth probe here, the Benke-Fried probe, that has a stimulation and the most distal LFP contacts. And we are doing micro channel recording 7 millimeters apart. So we measure the response here in response to increasing the amplitude of the stimulation in order for us to understand how much amplitude is required for this stimulation artifact to be handled effectively. So we did that. We took a whole bunch of measurements, and then we observed here with respect to the stimulation signal amplitude for this patient, increasing the current from 0.2 to 0.6 milliamps here for the microstimulation. That we see here that blue represents signal and artifact. So you can see that the clean signal is a very small amount, which is superimposed on the very large artifact. And so you can see here that you need, for this much, you have 0.6 milliamps, you need 20 millivolts extra dynamic range, which is 5 bits for linearity for your front end. And so we took that and we developed a range of techniques. Some of this is our work over here that is presented here, that we first used time to digital conversion using voltage controlled oscillator. And the idea there was, if you have a gigahertz technology that needs to sample a kilohertz rates, giga over kilo, that's a million, that's 2 to the 20. So we should be able to get 20 bits overall, minus some for the non-idealities and noise. So 14, 15 bits should be doable. So that was the idea. We were able to show 100 millivolt peak to peak. Here, it's also important to look into the impedance to prevent electrolysis. A lot of this work in related references does not really put much regard to the impedance. That was kind of based on comparison on Medtronic design from 2007, where they've shown chopper-based amplifier, which really is good, but you know, there is a lack of understanding, I think, generally in the community, that Medtronic uses DC blocking capacitors to afford a chopper-based amplifiers. So if you're dealing with 64 contacts, placing 0201 capacitors all over, it's not possible. So you need to have high impedance on chip. And so we provided that because, like I say, we look into all the clinical requirements for the application. Then we extended this to 200 millivolt peak to peak, included support for single units, and had to deal with the common mode rejection, because when you're dealing with very large input signal, then you need to have a large common mode signal tolerance. So that was an evolution of various technologies for the sensing to provide the need for ability for sensing. And then this is kind of the collection of modules. Each one of these components is essentially gone through a series of chip technology iterations. Each one of these also has gone to 2 or 3 iterations for assembly and compliance with packaging. It was a very large team of about 50 people distributed across collaborators. Clinical lead for this was UCSF. UCLA was responsible for technology, and then we have a number of collaborators, most importantly Lawrence Livermore and Boston Scientific, and other vendors that made certain parts to the spec. So this was the exhibit at DARPA 60, which is the 60th anniversary of the agency, where we're showing a demonstration here of responsive closed loop stimulation as compared to stimulation, which is shown in green, that is essentially open loop. And we also had a video of a patient for a clinical validation who experienced and declared that in presence of stimulation, severe anxiety was pretty much, resolved. And the patient said she felt normal, which was really amazing to hear for the mental health kind of indication as a proof of concept. When we do system verification and all the things that I like to say, this is all the things that we don't publish, but they're absolutely essential. We've developed a simple connector board, the motherboard, that is able to aggregate all of the components at various levels of maturity and various levels of assembly. So here you're looking into the Goldfinger framed module that can be verified, and then after the packaging, it can be plugged into a different place and then verified again. So here we have simulator of the neuromodulator, and we have here the interface for the actual hardware. So we can swap in and out the components as they come at different level of maturity. And this allowed us to verify all system components and full communication chain and control from a PC application here that had this external radio module, which could also be hardwired to the connector board. So this is all the modules in their final shape and form at the end of the 5-year program in 2019, featuring the test module with a custom developed data coil for communications, then aggregation module, which is a signal router, which we built in sufficient quantity for qualification, the vitamin pill sized neuromodulator with proper enclosure, and a whole bunch of other components like cables to connect the modules, PC application. And you can see here the evolution of this control module and verification that in itself was a 50-page report, 20+ point inspection to show various verification points for this technology. And this is pretty much where we ran out of time and ran out of money to qualify this. And I think it was a good decision, because further spending on qualification of such a system that is just a research level readiness wouldn't be necessarily the best way to go. This clearly can be repackaged into kind of a smaller form factor device that has a large electrode count and features all of the closed loop functionality. So these are the comparisons. We were aiming to get technology specified for IDE compliance. We have not cleared it for IDE through FDA, but we had all the documentation and history file. It was built on the principles and requirements for IDE compliance. We were able to achieve 256 electrodes with sufficient current for DBS as well as for the science community, with very low compliance voltage through a lot of innovation in the power, integrated power system, with full duplex technology. And ultimately, it was a bulky system with the way that it was conceptualized as modular. So we would like to translate this into system that's more practical and higher level of maturity towards therapeutic use. And we were limited here on the radio, which had a 4 megabits per second, with running out of resources on a microcontroller to do the MAC layer, which can be hardened into the hardware as well, and come up with generation 3. These are the references in the comparison table that I used, and going from where we are now, I think we envision a device that is not meant to be on an extracted brain like this, but it's a basically single body device in a single packaging that has then flexibility to connect up to 8 electrodes that can be used as temporary epilepsy monitoring implant. And once you have the ability to enter epilepsy monitoring, you can do stimulation for brain mapping. And once you're able to do stimulation, you can do clinical translation, and you can do translational science and provide enough evidence to the FDA to approve indication-specific studies. And it's kind of catch-22, because you first need to have a technology in order to get the results, but you can't get the results without technology. And so this would be the way really to kind of address all of that. Neurotechnology landscape is very large. I always am mindful of what else is being done to kind of justify any further customization and development. There is a large number of medical device manufacturers focusing on electrodes and sensing, stimulation, BCI. And so all of that kind of goes into decision making when we build our technology. So where we are now is we took technology developed for the DARPA program and created external device, which I call Cracker Stack, because this really is like a size of a couple crackers stacked up on top of each other as you're eating snack. So red layers have 64 contact generic omnetics interface stimulation and sensing. Yellow layer is the data management, and the green layer is the control, which is on an FPGA. And so we also have a user interface that allows us to do stimulation and sensing. We cleared this through our clinical engineering at UCLA and approved it through IRB for human subject studies. And we've been running recording and also stimulation studies in about 10 human subjects so far. And we are able to show how our technology here with proper packaging and shielding against power line provides a concordance, in this case with neon codon equipment, that you can see imperceptible differences in time domain on the left and frequency domain on the right, with the technology that we built that has far more ability for sophisticated stimulation waveforms. And we were also able to show, to the best of our knowledge, the first human subject walking around hospital room with a closed loop neuromodulation device. So those are the kind of results that scientists have been able to show with the place cells in animals, but has not been done in humans. So we are collecting all that data slowly but surely and preparing disclosure of our findings in a publication. Just to end on this kind of neurotechnology, I have looked into external devices that would be kind of class 2 devices, and this turned out to be one of the hobby projects that I did when I was on my sabbatical at Stanford in 2013-14 to work in semiconductors, and then I worked together with a neurologist and computer musician, Professors Parvizi and Chafe. And I built a neurotechnology that basically implements their algorithm for sonification of the brain. So you have frequency response on EEG between 2 and 20 hertz for epilepsy, for seizure detection, which is not in audible range. So that's where the computer music part comes in. So you do frequency modulation to audible range and do some kind of a algorithm to make it smooth so that you can put this through a speaker and hear the sound of the brain. So we called it originally brain stethoscope, just because you can put a stethoscope for the heart on this device and hear how the brain sounds. Now this is under Cerebell and the technology which has been modified, but based on these principles is in use in over 170 hospitals worldwide. And you can see why and how this actually works in a very simple way. So this is one sound of a person without seizure. So you should be able to hear that. That is just a background alpha activity at 10 hertz that you have a normal brain. And so let me show you a sound of a patient with seizure. So you can see that it's immediately recognizable, these kind of earthquake events, and very easily detectable. So we gave this kind of a training to a group of high school students and gave them a library of over 100 patient records, and they were able to score seizure outcomes better than trained neurologists looking into waveforms, due to simplicity of this technology that makes it really accessible for kind of outpatient monitoring. And so I would just like to end by saying that with a lot of public disclosures about artificial intelligence and neurotechnology, what are the social implications? I would really cross out these implications and say we have social responsibility, that before we can even discuss AI, we need to address medical needs. We have enormous medical needs in network scale disorders, and we gotta squarely look into that first. So I think with the closed loop neurotechnology, with a high number of contacts, we should be able to revolutionize non-pharmacological therapies for mental health and other indications like active memories and perhaps chronic pain. And then once we have those technologies already embedded, we can think about beyond medicine. But I would even then say we need to do gradual approach to look into mechanisms of cognition, memory, the quality of sleep, things like that, and be able to understand how we can provide treatment and augmentation of these functionalities with the technologies that we already have, so that we can overall assist and accelerate human learning and knowledge. And I think, you know, mixing brain-computer interfaces into the AI story, I think it's premature, it's naive, and simply speaking, technology and biology have fundamentally different operating principles that it's difficult to compare those worlds. But we have a lot of work to do, and I think medical focus has to remain for us. And just looking into the existing technology, I put this kind of a contrast, maybe a bit of a skewed analogy with old technology to say that we innovate so much in consumer applications and we do almost nothing in comparison in the medical space. So I think we can no longer implant telephone poles into people in the era of smartphone technology. We have a great opportunity now to combine modern brain-computer interfaces and big data analytics for medical devices as a service and miniaturized implants to revolutionize modern medicine and provide good quality of care for many people who need it. Thank you. That will be my presentation. I took longer than I had hoped, but I'm available to stay around and have discussion.
Dejan, thank you. It was very inspiring. We have time for questions.
Yes, please.
Yeah, Avi.
Okay. May I?
Please.
Okay. Yes, thank you. If I understand correctly, the closed loop is the same modality that you measure and the same modality you actually stimulate. Both are electrical sensing and stimulation. If, for instance, I look only at the sensing part, will you be able, with the arrays that you have, to actually map not only the points that you touch, but a much larger volume of the brain? And then maybe you would like to use another modality to actually do the stimulation. For instance, a focused ultrasound helmet, which might enable you to get in many, many other states or positions in the brain.
Yeah, multimodality is definitely a good way to go. I do think also that associated benefit of sensing is that we can also quantify at some level biomarkers of that could inform administering of medications. And I'm in no way saying that you know, medications should not be taken. This is just like a part of comprehensive care. This is applicable for patients who can no longer be helped by medications, and then we can perhaps by sensing inform at some level the dosing and response to those medications. I do agree that bringing more modalities such as optical technologies perhaps, or together with imaging, together with focused ultrasound, anything that helps us essentially achieve sufficient spatiotemporal resolution is definitely worthwhile. I think it really comes down to spatiotemporal resolution, that with electrophysiology, I think we are limited in spatial resolution, but we have sufficient temporal resolution. So if you have the ability to have enough contact points, then we would be able to some extent address the space. For indications like memory, that would be very challenging because of the specificity. So we'll have to see to what extent we can make inferences based on the limited number of access points. But I agree with you that one technology cannot do it all, and they all need to work kind of in a cohesion, and we should have good amount of analytical ability to aggregate and interpret this big data that comes from also observational behavior, other biomarkers, and understanding of other related indications that will need to be coming into the therapy outcome.
Thank you.
I have a very basic question regarding the nature of the signal and its propagation in the brain, not in the wires. Mm-hmm. Is it merely a matter of collecting time-space samples, like point samples, or can you also do the equivalent of beamforming because there's propagation? In other words, what is the range? When you sense at a point, are you sensing only the effect of 0.5 millimeter around you, or 10 millimeters?
Great question. So this comes to really kind of the classical aperture problem. So if you have essentially you start with essentially understanding that the density of neurons is about 300,000 per millimeter cubic. Right? So that informs you how large an electrode has to be in order to achieve certain level of specificity. So if you have a microwires that are maybe 50 micron diameter contacts, or maybe 30 micron, then you can maybe get about typically 2 to 5 single units that are in that vicinity, but it would be difficult. And at the same time, difficult to position them to the right places. But at the same time, you're also seeing all of the background noise and activity from the neighborhood, from all of the other local field potentials.
Yeah, this is basically my question. What is the range, the kind of reception range? In other words, if I could artificially activate something 2 millimeters away, would you sense it, or it's way too far?
Yes, yes. So millimeter scale, definitely you can sense. So the active end of the electrodes, if you look into the DBS electrode, we are talking about typically about 2 centimeters. And so you would be able through this kind of volume conduction to see the effect of stimulation itself that is coming through the brain. And brain itself, and this is one of the really interesting, I think, experiments, and what I'm trying to find out as well is, how does the brain look as a propagation medium?
Yeah, exactly.
Its own, right? So we understand wireless communication. We can do that model, Rayleigh fading and so on. But I tell, people that for the brain, when you're thinking about, for example, let's say in the communications, you come closer to a base station, you can drop the gain and you can afford more noise because your signal is so much stronger that you can still have high signal to noise ratio for reliable detection. When you're talking about the brain, nobody amplifies your brain signal. Your interferer is right in the band itself. And you need both high range and you have high sensitivity with low noise. So that becomes a really kind of different and tricky way to look into it. And I'm still appalled with how much people jump onto communications kind of receivers to build brain sensing technologies. Those are just two different problems. Yeah. But yeah, you do get millimeter, definitely several millimeters of response. The splitting of the rings of the electrodes gives you that ability to do current steering, to kind of get the field lines to shoot in a kind of beam forming way, but very crude, that you can direct the currents into the volume. People from the experiments clinically so far haven't found much merit. Like Boston Scientific has this 1-3-3-1 lead. You have like one ring, then you have three-way split, one ring. And so you can do kind of certain level of directionality. But I think part of that is also now a problem of increasing parametric space in the clinic. Like so far, we have been coming from the world of 130 hertz stimulation. Nobody questions the amplitude, the frequency, because someone determined that's the way it should be. And people dialing the knob more amplitude or less, right? So from the clinician point of view, when you tell them, now you can play a piano on the brain. I have, let's say, 8 stimulation engines and many keys. That's exactly the kind of analogy for this technology that we are using. And so what we have developed, at least, is the automated routine that exercises these different stimulation parameters with respect to frequency, amplitude, shape, and location, so that we can get stimulation response maps, so that we can understand how much of that is really volume conduction through the stimulation and how much is that really meaningful response to functionally connected network that got activated as a result of stimulation. And so that is kind of the data that we are collecting and trying to understand, which is not easy to understand, but it's a very interesting question overall to try to really figure out when do we need to stimulate where and how much to get the most effective clinical outcome.
Yeah, it seems that in brain terms, you're doing very coarse things. Because if you have the densities you talked about, obviously you're not affecting a single neuron or anything.
Correct. That's right.
Yeah. Now, regarding the propagation, there may be an interesting kind of self-contradiction argument that if you have very dense signal points and you claim to have a broad, large range effect, then the crosstalk among them has to be very high, the interference, then they should kill each other signal-wise. So you can't have it both ways somehow. You can't have very high density of things that are each have a large range effect.
Correct.
And they self-interfere terribly, so it can't. Okay, thank you.
Okay, any other questions?
I have one question, please.
Yes, Nado.
Professor Markovic, I've recently read your papers, your group's paper in TBcast published in 2019 about the artifact-free simultaneous stimulation and sensing. And I see that the way you train the artifact channel to cancel the stimulation artifact is by blocking part of the neural signal. First, you study the channel you study the neural signal, you get some statistics of the neural signal, and then you block it. And then you pass the training signal only to a normalized LMS algorithm. This is how you get an artifact-free channel. But my question here is, aren't the neural signals supposed to occupy all the headroom of the circuit? Otherwise, if it's not, we actually lose ENOB, or effective number of bits, at the output of the system. Can you please comment on that?
Yeah, so the neural signal does have to occupy, to be always present. The question is, how do we detect how much of the response is due to artifact that we can remove? And we have used the technique of measuring the response with respect to frequency and amplitude, because there is some nonlinearity there as well. So the best way you can characterize really the response is not by knowing the perfect shape that you are injecting into the tissue, but your response is going to be distance and amplitude and time dependent. So you really need to characterize for that at the reception points of interest, so that if you say, now I'm going to spend some time to understand the statistics of the signal itself, and then I'm going to have a response that is due to artifact of stimulation. There might be some additional activated behavior of the neurons due to stimulation that would be difficult to characterize, because we do not have, that's kind of blinded by the stimulation artifact plus that. And we have also looked into exploiting mutual information, that you take a multi-point measurement that allows you to have additional information from adjacent points to understand how the stimulation artifact reflects itself on the measurement point, so that you can then consequently remove that amount.
I see. So effectively, we actually don't get the full number of effective number of bits from our ADC. Let's say if we have an ADC of 10 bits, we would want the neural signal to be at least, that's entering the ADC, we would want it to be at least occupying half of the headroom, so that if we blocked it, we still have the other half of the headroom to pass the training sequence. Is this true?
We have more than 10 bits. So the ENOB, the front ends we built have 200 millivolt peak to peak full linear input range with a noise level and 2 microvolt or 7 microvolt RMS for LFPs and units respectively. So you do have plenty of headroom, and it is a linear range that you are not dealing with distortions, unwanted distortion of the additional distortion of the stimulation artifact. So you capture the full range and full content of neural signal and stimulation artifact. And if you previously know the statistics of the neural signal, you would be able then to do separation of the signal from the artifact.
So you're basically estimating and subtracting. You're not blocking anything.
Right, right.
Yeah, okay, that's...
A known technology, very old one from the inertial navigation arena, is using what you've mentioned in your lecture, a voltage to frequency converter, which gives you a lot more resolution. And that's a known technology.
Right, yeah, yeah. And there we also had a little bit of interesting, I think, dealing with the non-linearities of the components that we used to build these voltage control oscillators, which made us think, if we can do non-linearity correction on that, shall we do that in first to have a perfect linear range and then do the artifacts, or we can try to kind of do both of them at the same time. So those are kind of interesting, you know, discussions for which we just didn't have enough data to see how to do it. And now that we have access to the human brain, we can get some idea through the measured simulation responses to see how would be the next generation technology looking like with all that accounted for.
Is compressed sensing relevant to all this, to reduce power, bandwidth?
Yeah, so absolutely. So I think there is a different levels of that question. So we did look into this before, what we called EXG, which is non-invasive, like EEG, EMG, and EKG signals. And we determined that about 3 times compression would be practically possible with acceptable signal reconstruction outcome. A lot of time people, you know, put compressed sensing and they just basically say, hey, this kind of roughly looks like, but we looked into the reconstructed signal ratio with respect to the original golden standard, and we saw that even though these signals look very sparse, you really cannot compress them too much if you want to have full signal fidelity. Mm-hmm. And this is kind of competing between basic and clinical scientists who don't want to discard any data. For the epilepsy monitoring, for example, that I mentioned, you do 3-week recording, and it's about 3.6 terabytes of data for every patient. And people literally keep them on hard drives that they put on shelves. But if you look into how much information is there, it's like 1 byte, which is then ID of the channel 1 through 64 that needs to be surgically resected to remove the tissue. So that's kind of really large data mining problem. But in this case, like you can activate sensing periodically. You don't need to do always on sensing, right? You can say, For example, time constants for epilepsy are different than time constants for mental health, right? So if you're doing epilepsy, you may need to sample more often, but for mental health, maybe you activate sampling every 5 to 10 minutes, and then you sample for 30 seconds, and then again, and then after that, you kind of go to sleep. So you can do this kind of importance sampling at the time constants and interval epochs that are relevant for biological behavior, that we can kind of duty cycle the device so that we can get meaningful data.
And can you store in the body so that you can transmit at a fixed rate going out?
Yeah, that's also a great question that comes to the technology limitations of how much storage can we afford. And our position with that is that the best, and there is also the security aspect of it, the security of wireless data, right? When people come to hacking and things like that with the brain, you don't want to take any chance. And typical practical, comfort zone is that if you are within the arm's length of somebody, then that's kind of the safe distance, right? Nobody gets into your personal space. But we that's for that reason, we put the technology for wireless that is 3 to 5 centimeters away. So that's really even less than the arm length. And we put AES 256 on top of that. So it is almost like a bulletproof wireless link. And then it relies on the external technology like a helmet or a hat or something that's socially acceptable and inconspicuous, that can, when it's placed, it can then transfer data at full rate. So you can just, at that time that you say, now I want my data, you put an external device, you get full data downloaded with a secure link. And then if you are, based on that basically data collection, you want to do system identification and then decide based on that vast amount of data, you then decide, okay, now I understand how to program my online closed loop. And I'm going to determine, first of all, based on everything, which subset of channels do I need to record from, what features I need to record, and how does that map into control policy for the stimulation for closed loop. I would say, within this range of parameters, when I detect this, my algorithm is going to do closed loop. And at that time, you turn off the radio. You say, I trust this device is doing fine. It's doing autonomously what it needs to. You can still have some amount of history to say, give me few minutes of this or 30 seconds, whatnot, to show me how the algorithm actually worked.
Yeah.
But ultimately, you should be able to have an override of that and say, whenever you are charging a device through an external helmet, for example, or a hat, at that time, you're also uploading device to the cloud. Mm-hmm. And then clinician can take a look, or some big data analytics algorithm that would be automated would say to the clinician, hey, you have to take a look into this data because it seems like it's completely out of spec of what we originally anticipated. And then clinician would say, okay, now you have to come to a follow-up. And then you come to a follow-up for a clinic to do this kind of calibration routine again. So I think that way we can implement remote tracking of the patient's well-being every time they recharge the device. And then you kind of promote, just like you have on your iWatch or something like that, that you need to charge it every few days or so. You can enforce this to be charged every few days as well, so that clinicians get more data. So more data is then better. And then we can have these informed follow-ups rather than you come every 6 months, whether you need to or you don't. And if there are some catastrophic events in the meantime, too bad, we don't know, right?
Itai has a question.
Yeah, I wanted to ask about neuroplasticity. How can it, is it exploited in both sensing and stimulation?
Great. That's another great question. You don't want to overstimulate to kind of mess up with plasticity, but you do want to stimulate at a settling time constants that would promote successful neuroplasticity, right? So that, I think, would need to be included in the overall stimulation policy, but more of that would be really known with more data sets and more longer time. We just don't really look into that aspect, into long-term potentiation and depression to figure out how it works. But generally, I would say with therapy, less is more. And if we can invoke neuroplasticity and adaptation of a brain state, then the best way to do stimulation is to turn it off when we don't need it, right? And so just activate it only in cases where we need to go from one state to another, and then make sure that that state is eventually a stable state, right? And that's like with medications. If you think about it, you feel sick, you take medicine, and then after that you recover, you're okay. And ideally with neurostimulation, it should be like that as well, right? If we are able to promote repair, then the most successful outcome would be that you don't need to stimulate anymore.
Any other questions?
Yeah, I have another question, this time at an FDA level.
Yeah.
The FDA or someone insists on not on only lossless compression for imaging data or things like that. It's amazing that the same people will allow you to get away without recording everything that you did when you're actively doing something to a patient. It's a contradiction. I mean, don't raise it with them because you know what way they'll go. But I think it's funny to observe this, that you know, you'll record the terabyte, and they won't force you to record what you're doing autonomously when you're actually stimulating. It's very
even more scary is that they allow people to do blind stimulation. It's like a black hole, right? Blind stimulation without any feedback, right? There was an interesting article actually that was published by Josh McCauer, who is was I think clinical professor at Stanford, and he's now working with the New Enterprise Associates with as a venture capitalist. And he wrote an article basically asking a question, you know, is it better to talk to FDA early on in the process or after you're completely done? And his conclusion, at least from the data points he analyzed, was that do not talk to FDA early because they would give you more constraints than you should be dealing with. But you need to understand all of the safety requirements and then come to the FDA and say, yeah, I've addressed all of them. And I just developed my device the way it should be, instead of imposing undue burden on your own development to kind of delay the overall process. It was kind of interesting.
I think the ultimate test is the malpractice insurance premium.
Right.
If you can get insured at a reasonable rate, that's the test.
And that's what I think when I say this medical device as a service would be way to go, that you go to insurance and say, pay on an outcomes basis, right? Instead of paying immediately for a high cost of device that is amortized, you instead pay a payment plan over 7 to 10 years that if it's successful, then you continue that subscription because that means you have a value out of it.
In Israel, by the way, this was done last year. We have in Israel an annual budget of approximately $150 million, small country, for new technologies. And there's a committee that sits, you know, the demand is much higher. They prioritize and decide. And last year they did something special. There are these new medications that want, you know, $1 million per patient, things like that. So they struck a deal with them that they will pay them the 1 million, but only if successful.
Right.
Otherwise, they don't pay. And with a high risk in the sense of not succeeding, and they managed to pull it and got a lot more in by so doing. So no, that's medicine, but anyhow, same kind of idea.
Yep, I think that encourages fair competition, fair trade.
Yeah, fairness, and you kind of put the provider on the same side of the fence.
Right.
Because if he fails, he doesn't get anything. It's not that he tricked you into buying.
Right, right.
So, yeah.
Okay, I think that it's time to finish. Dan, thank you again for the very interesting talk, and more importantly for the great work, and it's very important also. So,
my pleasure. Thank you all. It was great talking to all of you. And like I said at the beginning, I understand this is a family time and dinner time, and I hope you know, I was able to provide meaningful content. Otherwise, you could have watched the movie or done something more interesting.
The alternative here is to watch the political news, and these days we don't want to.
That's right.
So, yeah. Okay, thank you very much. It was really interesting.
Thank you.
Thank you.
My pleasure.
Thank you.
Can I quickly ask a small question?
Yeah, go ahead.
So regarding the spatial resolution, the final goal is to stimulate specific cells, like to be able to stimulate like the play cells, or regarding memory, eventually it's more in a cellular level. So you think that at the end of the day, the product will be a lot of electrodes inside the brain, or?
Good question. I think the idea is really to stimulate the right places at the right times. It would be difficult to do at a cellular level, but it should be precise enough to have not just the right structure, but the right function as well. So we did at some point look into more than 100 images of the brain to study memory, and then we realized that there are, you know, millimeter scale or so anatomical variations that prompted us to take that into account in the electrode design, so that we can always achieve the same at least circuitry to be measured and stimulated. And so in memory particularly, the focus was on recording from the hippocampus and stimulation of the entorhinal cortex of the alveolar bundle, that white matter that really gives you projections into the hippocampus. So, and if you can furthermore do that with respect to phase of the theta rhythm, to really be looking into specific aspects of encoding or retrieval that occur at the peaks or troughs of the theta rhythm, then you would be able to really kind of, with a minimal disruption, synchronize the stimulation with respect to the natural rhythm of the brain when it comes to retaining new information or recalling the previously recorded information. So I think if we do those kind of global networks and the circuitry, we would be able to achieve certain measurable outcomes, but certainly wouldn't be able to get down to the level of individual neurons. I mean, it'll be nice if we can, but it's just difficult.
Yeah.
Do you know what the stimulation actually does? Does it synchronize things, kind of reestablish some kind of rhythm, like sign in the heart? Or what
so stimulation, people measure, does phase resetting. Okay. And so it's kind of enforcing the phase, really. It's almost like a shouting into the brain and disrupting the flow, but it would be nice to have kind of in the way that the brain already works as kind of the models of these kind of synchronized oscillatory behavior. And, you know, theta rhythm itself is interesting because people kind of discovered, oh, theta rhythm in rats is higher than in humans. Yeah, because you measure theta in rats when they are running, and humans when they are kind of under medications, kind of laying in bed. So that's kind of something that you need to kind of provide there based on the activity level. You have the assessment of the theta rhythm estimates. And then with respect to that, you are then trying to synchronize stimulation with respect to that.
So it's something between a pacemaker and a defibrillator in heart terms,
right?
Cardiac terms.
Right.
Okay. But you don't imagine it as a, like optogenetics tool that activates, you know, engram cells and stuff like that?
Oh, that will be wonderful. I mean, if we can do that, what I like to say is like anything that FDA has not already approved, we would need to probably add another decade. So for optogenetics, I think we probably would need to stagger that for 10 years later behind electrophysiology, because the technology in electrophysiology is still so outdated that I think there is still big room of improvement, that we can bring it, I think, responsibly through epilepsy monitoring to do a thorough study to see if there is a benefit of network stimulation for these other big indications. But I agree with you that accessing broader and finer grain features would be, you know, necessitating other technologies to come into play over time.
Yeah, continue.
I wanted to ask if you think that in the future you will also monitor like biological cues like I don't know, serotonin levels or dopamine levels to validate like what you said, that you have the app to say I'm happy now or stuff like that. So maybe that's another thing.
Yeah, I think we would definitely want to measure as much of biomarkers as possible. And ultimately, it comes down to function and behavior. In the device that we are doing with all this kind of brain stimulation and the piano kind of stimulation to see if the brain is well in tune, in the end, it really comes down to simple clinical feedback. I'm feeling better or I'm not. And then all of those complexities of technologies need to be abstracted away. But on that note, everything and anything that comes into a patient chart, right? You have a family history of that disease. Do you have any other problems? Do you have allergies? Do you have, are you taking any medications? All of that has to come into a comprehensive view, because you cannot compare two patients in the same way because they have their own kind of a history. And sometimes, you know, they are not feeling well and they don't necessarily give you correct information, or there would be other ways of kind of quantitatively assessing their overall well-being in addition to what they're subjectively reporting. And so I think we do need really to take a fusion of all those data modalities, even the heart rate, blood pressure, whatever is relevant physiologically. I think we need to account for that and include it in our overall analysis.
I suspect there's a smartphone with its camera today and a little machine learning can get you most of the supplemental information. The camera looks at the face, it knows whether the person is happy, suffering, whatever, with a little training.
Right.
Because really you need one bite, as you said, of this kind of information, not...
Okay.
Okay.
Yes, Dejan, thank you again.
Thanks again.
Sure. Thank you so much. Bye-bye. Have a good night.
Thank you very much.
Thank you.